McDougal, and I'm a freshman at the illustrious North Carolina Agricultural and Technical State University. And today I'll be walking you through my protein structure project. I've been assigned with the type 1 collagen, which is a structural protein that is an essential component of bone formation and comprises around 30% of the proteins in the human body. This protein provides flexibility as well as stability of bones, tendons, skin, and ligaments. Type 1 collagen is normally found in the extracellular matrix of animal cells and connective tissues, as well as muscle and bone cells. The basic structure of collagen is a triple helix that consists of 2 alpha-1 chains and 1 alpha-2 chains. For my model, I've depicted the alpha-1 chains with pipe cleaners that include pink and green puffballs, and the alpha-2 chain has orange and blue puffballs. The structure also includes 3 major amino acids. Glycine, which I've depicted with the small rhinestones. Proline are the pink and blue puffballs, and hydroxyproline are the green and orange puffballs. Methyl and hydroxyl groups are present in the hydroxyproline. Methyl is also present in the amino acid proline, and glycine includes the functional groups carboxyl and amino. Glycine is very simple and only has a hydrogen atom. That's why I chose to represent it as a small rhinestone. This makes it much smaller than the rest of the amino acids, as well as nonpolar and hydrophobic. Proline and hydroxyproline are nonpolar and hydrophobic because they contain methyl groups. My assigned mutation for this project is substituting glycine for a larger amino acid, resulting in a disorder called osteogenesis imperfecta. On my model, I'll be taking a larger puffball and putting it in place of the glycine or the rhinestones to show the mutation. Osteogenesis imperfecta is more commonly known as brittle bone disease. As a result of this mutation, the protein becomes less active because the large amino acid prevents the tight coil required to link the triple helix, which leads to an inadequate production of organic matrix bone, which is a key formation of bone structure. Based on the specific amino acid chosen to replace the glycine amino acid, the more severe the case of osteogenesis imperfecta is. Moderate cases are found when alanine is substituted for glycine. Mild cases include cysteine, serine, or glutamic acid substituted for glycine. And the severe and oftentimes deadly cases are a result of arginine, cysteine, valine, serine, or aspartic acid as a substitution for the glycine amino acid. When mutated, the side chain becomes much larger. Alanine, proline, and valine contain methyl groups, resulting in a nonpolar hydrophobic side chain. Glutamic acid and aspartic acid both include methyl and carboxyl groups, forming a negative charge. Cysteine only has a slightly polar charge and is hydrophilic. Lastly, arginine is basic and positively charged because of its amino, methyl, and carbonyl functional groups. The mutation will result in a peptide disruption because the bonds will be unable to accurately form due to the disproportional size of the substituted amino acid and the center of the helix. This mutation will also disrupt the repeated amino sequence glycine, proline, and hydroxyproline. In the normal structure, glycine creates hydrogen bonds linking the amino group to the carbonyl group and the adjacent protein, resulting in the structure of the triple helix. Proline and hydroxyproline form peptide bonds that allow them to fold into the helix. Now, while I did enjoy this project, I did run into a few limitations. As a college student, and especially as a freshman, I have a lack of transportation as well as a lack of financial stability, making it difficult to go to stores and find various products to successfully do this project. Another large limitation to this project was the fact I had to rely on my own knowledge, and with the limited references given, I had to gather the puzzle pieces to form a sound conclusion to answer each aspect of this project to the best of my ability. Lastly, time management was a limitation to this project. Although we had ample time to complete this project, juggling other class projects, essays, and tests, and studying made it difficult to start this project right away. There was also an issue with time management as I completed this project. Some components of this project came more easily to me, and others I spent time obsessing, which led to a drawn-out process of completing the project as a whole. I appreciate your time. I hope you enjoyed my project. Thank you.